second type of the qualitative hemoglobinopathy, and the second type is HbC disease. Now this HbC disease, it occurs due to the amino acid substitution in the sixth position of the beta globin gene, which causes the abnormality in the beta globin gene. Amino acid substitution at sixth position of beta globin gene. Now what happens? Which amino acid is substituted? We will be discussing now. What happens that lysine replaces the glutamic acid, which is already present in the healthy beta globin gene, but in we are talking about the abnormality, we are talking about the HbC disease. So lysine replaces the glutamic acid. This is a very important point. Due to this replacement, the HbC becomes less negative and its movement towards the anode becomes slow. So it moves slowly. HbC moves slowly towards anode. We know that anode is positively charged. So due to the replacement, it becomes less negative, so the movement towards the anode is slowed down. Slowly towards anode. And at the last, we will discuss about the symptom. It shows one symptom, which is the chronic hemolytic anemia, which we have discussed it before. Now this completes our HbC. Now moving on the HbC, which is the last topic of this lecture. Now, what is the HbSC disease? The HbSC disease, it is also the Hbc Sickling disease, and it contains the abnormal 2 beta globin chains, and these both are abnormal. We know that these both beta globin chains are abnormal in the case of HbC. Now, what is the HbSC disease? HbSC is also a Hbc Sickling disease, and it contains the 2 mutant beta globin chains, which are abnormal, but being abnormal, these are different from each other. Their abnormality is different from each other. So it is also a Hbc Sickling disease. Contains two abnormal beta globin chains different from each other. This is its first point. Now discussing the second point of this HbSC disease. second point, it is that it is said to be doubly heterozygous disease. What is meant by the doubly heterozygous disease? And the second point is, it is said as to be doubly heterozygous. Now, what is meant by the doubly heterozygous? This HbSC consists of the two beta globin chains that are abnormal. We know that. But this consists of the mutants that are found both in the HbC and HbS disease. Now, explaining the second point, it is designated by the name doubly heterozygous. Now, what is meant by the doubly heterozygous? We know that it can consist of the two abnormal beta globin genes. So it actually contains the two mutants, and those two mutants are found both in the HbS disease and also in the HbC disease, which means that it contains the mutant of the HbS and HbC disease. So both mutants of HbS and HbC disease are found in the HbSC disease. That is why it is said as doubly heterozygous. Both mutants of HbS and HbC are found in this disease. Now this was the second point. Now moving on the third point. In this disease, what happens that fluctuations occurs. Fluctuations in the hemoglobin levels occur. Which means that sometimes the number of hemoglobin or level of hemoglobin mounts so high, and sometimes the hemoglobin levels decline to a low number. So, level of hemoglobin rise and fall. And talking about its symptoms are not so severe than HbS. Now, what this line meant? Symptoms are not so severe than HbS, which means that the symptoms which HbSC shows, the symptoms are not severe that are actually shown in the case of the HbS. Means its symptoms are milder than the HbS. So this concludes our both three types of the qualitative hemoglobinopathy, and hope you have understood it well. Last but not the least, don't forget subscribing MS lectures. Thank you.